And I'm back. So we're talking about evolution and the driving forces that lead to evolution. So evolution is not only possible, it is unavoidable. Evolution is going to occur. It's organisms are changing. Now there's lots of organisms on this planet that don't seem to be evolving. And well, they look exactly like they did 10 million years ago. Yeah, and if you look at them really closely, they don't look exactly like they did 10 million years ago. They look really, really close, but not exactly. So evolution is going to happen. In real populations, allele frequencies will inevitably change over time because they are affected by so many selective forces. Again, here's a guy climbing a mountain. Why did you climb the mountain? Because it was there. Well, I got more important things to do with my life than climb a mountain. Scientists can test evolution. We do that by measuring the allele frequencies that do or do not change from one generation to the next. In this hypothetical population of ferrets, we have the D allele, which produces dark color. We have the capital D. We have the lowercase d, which produces light color. And then we have the intermediate here, which, if you can see this with a little bit better color quality, you can see that this ferret is darker colored than this ferret. And what we can do is we take this population of ferrets and we count the numbers of big D's and the numbers of little d's. And we come up with an allele frequency for the dominant allele and the allele frequency for the recessive allele. We can then, using the Hardy-Weinberg equilibrium, we can predict a hypothetical situation in which the allele frequencies do not change between generations. We have the ancestral here. The Hardy-Weinberg principle calls for random mating, no migration, genetic drift, mutation, or natural selection. None of these things happen, and this doesn't happen. Then the Hardy-Weinberg frequency says that the alleles, one generation and 50 generations later, we have the same allele frequencies. However, what we know to happen is that this is rare. This is generally the way things happen. We have snails here. We have an ancestral frequency of light blue, dark blue, and red. Individuals have low reproductive success due to some environmental change. And then we have genotypes with low reproductive success become less common. We went from a majority of this light blue to the light blue just barely existing. And we went from these two who were in the minority to now make up almost the entire population. Again, using allele frequencies. The Hardy-Weinberg principle relies on certain assumptions. Again, random mating, no migration, genetic drift, mutation, or natural selection. Again, this is theoretically possible, but it doesn't happen very often. And the big key is, among most mammals, mating is non-random. But again, here's the breakdown of how the assumption of the Hardy-Weinberg principle need to be met. Understand, what I want you to understand from this is, again, random mating. Each female is just as likely to mate with this male as with any other male. And that male is just as likely to mate with that female A, as any other. It's completely random. And that's an assumption for the Hardy-Weinberg equilibrium to work. Again, natural selection and mutations do occur. Random changes in allele frequency are common. Individuals rarely mate at random. And there's always some level of migration between populations. So the things that we must assume to be true for the Hardy-Weinberg equilibrium to happen don't happen. So what happens is natural selection. There are 3 different types of natural selection, and we're going to talk about, I want to look at something right quick. Okay. So, and they are directional selection, which is what you're seeing here, disruptive selection, which is what you're seeing here, and stabilizing selection, which is what you're seeing here. So let's talk about each one of those. Directional selection. These are what are called peppered moths. They have been used for 100 years to talk about evolution. There are three color phases of the peppered moth. There's the light one, there's the medium colored, and the brown one. And in a normal forest where there are dark colored trees and light colored trees, these will look this way. There will be a lot of medium colored ones, a few dark ones, and a few light ones. If you remember the bell curve we drew when we were talking about this in a previous chapter. But what happens when the population or something happens to the trees? Now, historically in England, before the Industrial Revolution, this is what you saw in the forest, because some forest trees were dark, but some forest trees were covered with a plant-like moss type thing called a lichen. And the lichens tend to be sort of a pale gray color. So when this bug landed on the lichen, it couldn't be seen very well. But if this bug landed on the lichen, it was seen. The birds ate it. And then this one could sort of fly between the lighter trees and the darker trees and not get caught as often. So that's why you had this nice bell curve. But then the Industrial Revolution came, factories start to be built, human beings start to burn a lot of coal, that threw a lot of coal dust in the air. Coal dust settled on the trunks of the trees and killed all the lichen. So over history, over a few decades, the trees began to all get dark. The dark trees were dark, and the trees that used to be covered with the lichen are now dark too. So these stand out a whole lot. These stand out more than before, but these now are still well hidden. So the number of dark ones starts to go up. The number of this and the number of this starts to go down. So you see our curve is moving to the right. That is directional selection. Disruptive selection, again, talk about snails here. The light and dark rocks allow for light and dark snails to survive and produce more intermediate color snails. Over time, the medium snails become less frequent in the population. We start off with a set of colored rocks that were intermingled. There are all kinds of different colors. And we see, again, these can get on the light rocks, these can get on the medium rocks, these can get on the dark rocks. But as we have a change and we start to see lots of light colored rocks and lots of dark colored rocks, our medium model disappears because these have a place to hide, these have a place to hide. But because we no longer have this complete sort of, rainbow's a bad word, but rainbow, we have dark, medium, and light rocks. All of these are able to survive. But as we simply start to get a mix of light and dark rocks, the medium one now is disappearing. And stabilizing selection. Health problems allow medium-sized babies to survive and reproduce more than their very large or their very small babies. Over time, medium-sized babies become more frequent in the population. So we start here. And what you will notice here is the number of individuals doesn't change, but the ones that fall in this area is getting bigger. You'll see your bell curve is narrowing. You know, this bell curve is this wide, whereas this bell curve is this wide. So it is, the population is stabilizing. Why are harmful alleles sometimes maintained in a population? Actually, sometimes harmful alleles can stay, and it gives us what is called the heterozygote example. An example of the harmful allele strategy is sickle cell disease. Now, everybody has heard of sickle cell. Everybody knows that sickle cell anemia is it's a disruption in the way red blood cells are made. When I was taking junior high school back in the 1970s, we were told that only African peoples got sickle cell. Only, and again, no offense, black people. We now know that's not true. Now, yes, sickle cell affects black folks much more than it does others, but because of the mingling that has gone on between Africans and Europeans and Africans and Asians and Africans and Native Americans, that the alleles have moved around, and now, yes, there's white folks out there that have sickle cell anemia. But what is this so-called heterozygote advantage? Okay, a person who is, and again, y'all remember, I don't like the word normal, but a person who has normal blood has 2 good genes for red cell formation. A person who has the sickle cell trait or is a sickle cell carrier has 1 good gene, 1 bad gene. The person who has sickle cell anemia has 2 bad genes. The one with 1 good, 1 bad, that's the heterozygote. If you have sickle cell, you are homozygote recessive. If you have, again, normal blood, then you are homozygote dominant. So what is the heterozygote advantage? The heterozygote advantage is this. In this area of the world where sickle cell comes from, you see the green area, that's the frequency of the allele. You see all of these other colors ranging for the sickle cell trait. The hash lines here are the distribution of malaria. And here's what we have discovered in the last 50 years. The folks living in this area right here, where the frequency of the sickle cell trait is most, the people who have the good blood, 2 good genes, they get bit by a mosquito, they get malaria, and they die. The people who have sickle cell suffer the effects of sickle cell anemia, and they die. But the folks who are the heterozygote, 1 good gene, 1 bad gene, they don't get sickle cell, and they also appear to have a natural immunity to malaria. By having lots of those intermediates, a lot of the heterozygotes, when 2 heterozygotes make a baby, they can produce all of them. 2 heterozygotes make babies. Theoretically, 25% of their babies will have 2 good genes, 25% of their babies will have 2 bad genes, but 50% of their babies will have 1 good gene, 1 bad gene. That is the heterozygote advantage. Again, this is the breakdown of the sickle cell trait. Here, the D, high susceptible to malaria. Here's the sickle cell disease. Low chance of sickle cell and low chance of malaria and no sickle cell disease. Again, 25%, 25%, 50%. Sexual selection. Again, ladies, you're the ones who makes the choice. Why are these 2 rams butting heads? Because there's some lady off camera over here who's wanting whichever one of these can butt heads the hardest. Why are these 2 rhinoceros beetles fighting? Because again, there's a female sitting over here. Sexual selection is the force that favors traits that apparently reduce survival. Again, I don't know how good that is for somebody. I mean, your brain is right there, and you're banging your head against each other. But sexual selection, we build complex nests, flash showy plumage, butting heads, all of that. That appears to waste energy. But what it does is it gives the female an opportunity to select who, in her mind, will be the best mate. Again, there's the rams, there's the 2 rhinoceros beetles. This is called a weaver bird, and he's weaving a nest here. He's taking all these grass blades, and he's weaving a nest on the other side where you don't see it. There'll be a hole for the female to go into. And ladies, you'll get a kick out of this. This bird, there's a female again. She's sitting in a tree over here somewhere, and he's doing this for her, and she's watching him make that nest. You know what she does if she likes the nest? She flies down in there and goes in it and moves in. You know what she does if she doesn't like the nest? She comes right up here and lands and cuts all that grass and drops it on the ground. She don't like it, but she ain't gonna let no other girl move into it. She's gonna cut it down, and she's gonna make this bad boy do it all over again. And again, it is the female that is always making the choice. That's a bird of paradise. You see all this showy plumage. This bird of paradise has all these plumes and all these feathers to attract females. But what it also does is it makes it really hard for this guy to fly. They don't fly real well. So they have a problem sometimes eating. There have actually been cases in history where the development of things like this have actually led to the extinction. There is a species called the Irish elk that is extinct, and it is theorized by many, many researchers that part of the reason the Irish elk went extinct is because the species kept selecting over generation after generation after generation for bigger and bigger and bigger antlers. And eventually, the antlers got so big, and again, this is a theory because we don't know, but the theory is that the antlers got so big that they were so heavy that the males with the biggest antlers weren't able to get into the position to make baby deer, baby elk anymore. And the females continued to choose the biggest antlered males as their prospective baby daddy, but the baby daddy couldn't perform because he couldn't get in position because the antlers were so huge. You only have to do that for a few generations, and then your numbers go down, and then you introduce human beings with spears, killing them, and again, these bucks with the great big antlers couldn't run very fast because their head weighs, as much as the rest of their body does, and humans were able to catch them, and we, between the giant antlers and lack of reproduction and human activity, the Irish elk disappeared. Mutations cause evolution to occur. Again, back to the pictures of the antibiotic. Here's the bacteria as the way it originally appeared. Here we have time passing, and this one mutation occurs. The antibiotic was designed to go after this. This, the antibiotic doesn't affect. So with no antibiotic absent, we keep this same sort of ratio because this is always going to be, even though, yes, it's going to produce more, but the ratio of this to this is going to remain the same if the antibiotics are not there. If, however, we put the antibiotics into the mix, these are going to start reproducing. These are mostly going to be eliminated, and we're going to change the ratio of red to green. Genetic drift is simply sampling error, sometimes by random. Only some individuals in a population reproduce, which causes a change in allele frequency purely by chance. Again, we're looking at picking marbles out of a jug. We take 10 marbles out of the jug. We originally have a 50 to 50 ratio. We take them out this way. We put them back in our jug. Our jug that was originally 50-50 is now 70-30. We take 10 more out. We now have a 9-to-1 ratio. We put them back in. Now we have a 90-to-10, and we take 10 out. We now have 100% the gold, none of the little purple. And here's the thing about genetic drift. Once these purple ones have disappeared from the population, they're gone forever. Unlike mutation, which increases diversity, genetic drift tends to eliminate alleles from a population. Again, once it's down here and we've got them gone, they never come back. An example of this can lead to something called the founder effect. The founder effect is where a severe reduction in population has led to a removal of alleles from a population. This is a baby and his mother that is a member of the Lancaster Amish population from the area around Lancaster, Pennsylvania. If you look at the entire population of all the Amish peoples in the eastern United States, you have a genetic diversity. You have this genetic diversity, and you'll notice there are lots of different colors of these little alleles. However, in Lancaster, Pennsylvania, the population is isolated. Nobody's moving in, nobody's moving out. I forget, I read this in 25 years ago in genetics class. Seems like there's like 9 families that are living in this area, and all of those 9 families for the last 120 years have been breeding in amongst themselves. And what has happened due to that small number of population, only 3 of these alleles are left. And if the blue allele and the red allele and the yellow allele, or excuse me, blue and red and yellow, show up in a baby, that baby will have this condition right here, which I knew the name of it and it has completely left me. I was hoping the next slide would tell me, so don't worry, you won't have to know what it is. I want to say it's Tay-Sachs disorder, but I don't think that's right, so don't write that down. But ultimately, if this baby gets all 3 of these alleles, it's going to develop a particular type of dwarfism. And if you look at their hand very closely, 1, 2, 3, 4, 5, 6 digits. It produces a particular type of dwarfism and something called polydactyly. And again, does this happen in this population over here? Yes. But it happens in this population with much greater frequency. And when those things start to happen with greater frequency, it is referred to as the founder effect. Another example of a type of genetic drift is called a bottleneck. Again, here's my original genetics population in the cheetah. Had 25 different alleles for some particular gene. When human beings decided that it was the coat of the cheetah looked better on humans than it did on the cheetah, and we started killing a bunch of cheetahs and making coats and scarves and hats and gloves and all that stuff we made out of cheetahs. 22 of those alleles were lost. We're left with only these 3 alleles. Now, as the population begins to rebound, because we are protecting cheetahs now, and the population is rebounding. It's not rebounding very quickly, but it is rebounding somewhat in Africa now. But we only have these 3 left, and that is referred to as a bottleneck. At some point, you've put something in a bottle, you put pebbles in a bottle or jelly beans in a bottle or something, and you turn it over and try to turn it out, and they sort of hang, and only 2 or 3 or 4 will fall out. That's called the bottleneck effect, because these genes are all in here, but when you turn it over, only these 3 fell out. Non-random mating. Again, for the Hardy-Weinberg theory to hold, mating must be random. Well, it's not. Mating is rarely random. Sexual selection and artificial selection tend to concentrate alleles locally, which affects allele frequency. There are other factors: geographical, temporal, behavioral. All of those things can play a role in making mating non-random. Gene flow is where existing genes move into an area where they haven't been before. Maybe there was always, I don't know, a river or a stream separating 2 populations, and the river dries up and they're able to cross paths again. This shows that you've got these lighter colored rabbits, these darker colored rabbits, and they're isolated populations. But what happens when this little rabbit decides to jump over here and join this group? The allele frequency in this population is going to change. And remember, what is the definition of evolution? Changing allele frequencies over time. This population has evolved because the only thing that happened was this person or this rabbit's alleles moved their gene flow. Evolution, again, is altered allele frequency, as I've said, for Hardy-Weinberg equilibrium to happen. You have to have certain parameters. And if you'll notice, we did the Hardy-Weinberg up here with the group of collective ferrets. One of the things about those ferrets that were studied in the Hardy-Weinberg equilibrium is they were the only known group of that particular ferret on the planet. They were only found in one place, and they had sort of this communal living arrangement where there was true random mating, and it held. But in most instances, the Hardy-Weinberg equilibrium does not work. Again, we've got the mutation here that causes natural selection. We have mutation, we have genetic drift, non-random mating, and then lastly, migration. So all of these things are ways that changing allele frequencies happen. And if you want to look at these numbers, you see here the 41 and the 31 are about the same, but here, you had a group that was 24, but now over here you've got a whole new one that doesn't exist over here. Here you've got the same bunch of alleles, but the frequency has changed. Again, the frequency is changing all over the place. Bacterial evolution goes hog wild on the farm. Farms where animals such as hogs are continuously fed antibiotics have become breeding grounds for antibiotic-resistant bacteria, not only in the pigs themselves, but they also tend to take up these antibiotics, and we get some of those effects as well. So in the control group, the number of resistant genes detected was less than 40. Farm 1, 70%. Farm 2, almost 90%. Farm 3, 80%. So what's going on? These farmers are feeding antibiotics to their pigs. What's happening? Like the picture showed a while ago, the ratio of resistant genes to non-existent genes, the ratio of resistant alleles to non-resistant alleles is being affected by this use of antibiotics. If you weren't using the antibiotics, that ratio would have remained the same. So why do farmers do this? Why do they feed their pigs antibiotics? To keep them healthier and make them grow faster so you can get them to market quicker. DNA analysis reveals that hog manure, as well as surrounding compost and soil from farmers using antibiotics, had more diverse resistant genes than did soil from the forest. So basically what they were doing is they were feeding it, they were looking in the areas where the hog manure was being placed. The farmer is collecting the manure, he's using it as fertilizer, possibly he has a dump site, I don't know, there's an area where a truck comes and collects it, but whatever. But the soil in and around the area where the manure was had a much higher percentage of the resistant genes than did the forest just, a mile away. Because why? Because all of that hog manure with the antibiotic residuals was in there. Again, not changing the bacteria, changing the ratio of resistant versus susceptible. And that is the biggest thing that people don't understand about antibiotic resistance. They think that the use of the antibiotic is changing the bacteria, and it's not. It's changing the ratio of resistant to susceptible. Normally, there's 99% susceptible and 1% resistant. But you start using antibiotics and you start just dumping them into the soil, those ratios start to do this. And if they get to the point where they're this way, we now have a bacterial infection that is no longer treatable by standard antibiotic therapy. If you have any questions, please contact me. trussell@uaptc.edu, or you may also reach out to me from the contact tab course messenger link within Blackboard itself. It's Friday where I am, so everybody have a good weekend, and I will see you next week.